Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 1937 marked a watershed moment in biochemistry with the elucidation of what Adolf Krebs termed the citric acid cycle, a metabolic pathway central to the energy production within all aerobic organisms. Before delving into the intricacies of Krebs' groundbreaking work, it is essential to understand the intellectual landscape that paved the way for this discovery. The field of metabolism was in its nascent stages, grappling with the complex interplay of biochemical reactions that constitute life. Early 20th century scientists had begun to unravel the processes of glycolysis, the breakdown of glucose, thanks to the pioneering work of Otto Meyerhoff and Gustav Mten, amongst others. However, the subsequent fate of the products of glycolysis, the compounds that held the key to energy production, remained a mystery. Cellular respiration, the process by which organisms extract energy from nutrients, presented a significant challenge. Researchers knew that oxygen was essential for this process, but the exact chemical steps were unknown. This was the intellectual vacuum into which Krebs stepped. Furthermore, the prevalent theories of the time struggled to adequately explain the observed rates of oxygen consumption and carbon dioxide production in living cells. Previous research on muscle metabolism, led by Archibald Hill, emphasized the importance of oxygen uptake. Yet a comprehensive understanding of how oxygen fueled the intricate dance of biochemical reactions remained elusive. The concept of a cyclical metabolic pathway was itself a relatively new concept, with the earlier work of Albert Szent-Györgyi demonstrating the importance of dicarboxylic acids in cellular respiration. Szent-Györgyi's investigations on the oxidation of succinic acid, fumaric acid, and malic acid in pigeon breast muscle provided crucial clues. However, many essential elements were missing. Krebs, drawing heavily from Szent-Györgyi's work, sought to build a more complete and coherent picture of the process. He recognized that a crucial component of cellular respiration had yet to be identified. Krebs' pivotal contribution, published in 1937, unveiled the cyclical nature of the metabolic pathway. The work provides a detailed description of how the cycle operates, initiating with the condensation of acetylcoenzyme with oxaloacetate to form citrate. This crucial step marked the beginning of the cycle. The citrate molecule undergoes a series of enzymatic reactions, each meticulously described in Krebs' published work, leading to the eventual regeneration of oxaloacetate, thereby allowing the cycle to continue. The paper meticulously outlines the sequential steps: the decarboxylation of citrate, the production of alpha-ketoglutarate, the conversion of alpha-ketoglutarate to succinylcoenzyme A, the oxidation of succinate to fumarate, the hydration of fumarate to form malate, and the final oxidation of malate to regenerate oxaloacetate. Crucially, Krebs identified the key intermediates within the cycle, including citric acid, alpha-ketoglutaric acid, and succinic acid, and demonstrated how these compounds were transformed by specific enzymes. Furthermore, Krebs correctly identified the release of carbon dioxide at two distinct points within the cycle, a significant observation that helped explain the balance sheet of cellular respiration. The cycle also includes the generation of high-energy electron carriers, like reduced nicotinamide adenine dinucleotide, frequently written as NAD, and reduced flavin adenine dinucleotide, often written as FAD2, which are the crucial sources of the electron transport chain. The work included the elucidation of the enzymes catalyzing each step, providing a clear biochemical roadmap for scientists to follow. Krebs also demonstrated that the cycle was not merely a theoretical construct but a functional reality in living cells, by using tissue slices and measuring oxygen consumption and carbon dioxide production. The paper thus provided a highly detailed overview of the citric acid cycle and the interrelationship of the steps involved. The direct impact of Krebs' work was immediate and far-reaching, fundamentally altering the understanding of cellular energy metabolism. It provided a theoretical framework and specific biochemical reactions for understanding how organisms extract energy from food. This knowledge, in turn, quickly opened new avenues of scientific inquiry. Researchers immediately recognized that the citric acid cycle was a central component of all forms of aerobic life. Krebs' rigorous experimental approach, including the use of tissue slices from pigeon breast muscle, provided a model for future biochemical research. The work laid the groundwork for understanding metabolic diseases and ultimately formed the basis for the design of various medications. The understanding of the citric acid cycle provided a paradigm shift in biochemistry, similar to how the discovery of the double helix reshaped genetics. Later, Krebs' work was used and expanded by other scientists. Albert Leininger, for example, further elucidated the role of the mitochondria, the cellular organelles where the citric acid cycle resides, and contributed to our understanding of the electron transport chain. The discoveries made by Krebs and others paved the way for the development of metabolic pathways beyond the citric acid cycle. The work spurred research into other metabolic processes like fatty acid oxidation and amino acid metabolism, all of which were understood to be intimately linked to the citric acid cycle. The cycle's discovery has found application in practically every branch of biology, from medicine to agriculture. The principles elucidated by Krebs continue to underpin the study of disease and the development of new therapeutic interventions. Knowledge of the cycle is essential for understanding conditions such as cancer, diabetes, a multitude of genetic disorders. The reception of Krebs' work within the scientific community was generally positive, although initial acceptance was not instantaneous. Critics, while acknowledging the significance of the findings, often questioned the precise details of the cycle, as well as the exact sequence of reactions. It is important to note that some of the specific steps and their precise enzymatic mechanisms were refinements to the cycle that came later. Subsequent research clarified aspects and deepened the understanding of its regulation. The work's importance, however, became quickly apparent as other scientists began to validate Krebs' findings and apply his framework to study various metabolic processes. The initial skepticism was gradually replaced by widespread acceptance. Those who initially questioned aspects of the cycle soon came to acknowledge the transformative power of Krebs' insights. The research of Albert Szent-Györgyi indisputably influenced Krebs' work. Szent-Györgyi's earlier studies on the oxidation of dicarboxylic acids provided a critical starting point for Krebs. Krebs built upon Szent-Györgyi's observations concerning the roles of succinate, fumarate, and malate in cellular respiration. He expanded their work, seeking to understand how these compounds fit into a larger, cyclical process. The early work of Otto Warburg, focusing on glucose metabolism and cellular respiration, also provided broader context. Although Warburg's primary focus was on aerobic glycolysis, his research helped to lay the foundation for understanding the fundamental principles of cellular energy production. Krebs' work, in turn, profoundly influenced subsequent generations of biochemists and biologists. The discovery of the citric acid cycle was foundational to the development of metabolic research. It directly contributed to the understanding of the role of mitochondria in cellular energy production, a pivotal step in the development of modern cell biology. The cycle's discovery spurred research into the electron transport chain, which generates a proton gradient across the inner mitochondrial membrane, used by ATP synthase to create adenosine triphosphate, often written as ATP, the universal energy currency of cells. His work provided the key to understanding how energy is produced from the food we eat, enabling researchers to understand the intricacies of cellular fuel production. The discovery of the citric acid cycle has influenced multiple fields, and it has continued to shape our understanding of the fundamental processes of life. The legacy of the 1937 paper remains a testament to the power of insightful experimentation and the enduring pursuit of scientific truth.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.